Cells of the adaptive branch of the immune system. B cells and T cells are cells of the adaptive immune system. They make specific antigen receptors. They require help from macrophages and dendritic cells, which are antigen-producing cells. Those are cells that bridge the innate immune system and the adaptive immune system. T cells express T cell receptors, or TCR, on their surfaces. So here's our T cell down here. Here's the TCR receptor. The TCRs do not bind directly to the pathogens. TCR can only bind to antigens presented to them by antigen-presenting cells, which is here, the APC. And like we said on the slide previous, APC, which includes macrophages and dendritic cells, have molecular cups called MHC proteins, right over here, multi-histocompatibility complex proteins on their surfaces. They hold antigens in a way that lets the T cells survey them. Right, so here's the pathogen, comes over here, and then it's presented, the antigens over here from the pathogen so the T cell can see them. This interaction is initiating an adaptive response. The APCs over here, it engulfs and processes the antigens, right? It takes the pathogen in, processes it. You've got the antigens over here, and it presents them in the MHC. Now, if a TCR binds to an antigen presented by MHC, the T cell becomes activated, matures, and divides. So here's our antigen here, that little yellow guy here, that's what the T cell is seeing. And then once it sees it, it's activated, matures, and divides. The two most important classes of T cell are the T helper cells and the C cytotoxic cells. T helper cells control and coordinate the adaptive immune response by releasing signaling proteins called lymphokines. Approximately 25 different T cell lymphokines have been identified. There are many different types of T helper cells found in different parts of the body and producing different proteins. Different types of T helper cells release different sets of lymphokines. Other cells have specific receptors for each lymphokine. When lymphokines bind to their specific receptors, the receiving cells respond by turning on or off. Each type of pathogen causes T helper cells to respond by releasing different lymphokines, making responses very specific. For example, bacteria will cause T helper cells to release proteins that tell B cells to make antibodies. A virus will cause T helper cells to tell killer T cells to kill virus-infected cells. Nematode worms will cause T helper cells to activate eosinophils, which are cells of the digestive tract that can kill the worms. Here's a few examples of lymphokines, right? We said there's 25 identified so far, along with their specific signaling role in the immune response, and they're shown here. So the lymphokines over here, and interleukin here, that's abbreviated as IL, okay? So we didn't write it each time. So here's what makes them, and here's what receives them. So if you'd like, pause this and just look at it for a second, then you can move on to the next slide. It is important to turn the immune system off after a pathogen has been eliminated to avoid chronic inflammation, right? That's one of the ways of killing pathogens, but you don't want it to go on forever. T helper cells also release lymphokines that downregulate, which means turn off the immune response after the pathogen has been eliminated. The T cells that downregulate are called T suppressor cells because they suppress the response to a particular antigen. T cytotoxic cells, also called killer T cells, kill virus-infected cells or cancerous cells by releasing chemicals that destroy their membrane or cause the target cells to undergo programmed cell death. The name describes their role. Cyto means cell and toxic means poisonous. T cytotoxic cells need at least two signals to kill. Their TCR is bound by a foreign antigen on the surface of the target, and they have a lymphokine signal from T helper cells. Here's what we've been talking about. We have our target pathogen over here. The APC engulfs it, presents the antigens to the T helper cell. That releases infokines. This then tells a T cytotoxic cell to release chemicals to destroy the membrane of the target. In addition to T cells, the adaptive immune system has an additional antigen-specific cell, the B cell, right? We've gone the T cells, now we're talking B. B cells produce antibodies. In a healthy vertebrate, antibodies do not bind to self structures, all right? This is part of the vertebrate. Yet B cells make antibodies that are ready to bond to any foreign structure or non-self structures. During surveillance, each B cell produces a unique antibody that it displays on its surface. Most B cells participate in surveillance only. They circulate in the blood and the lymph and are never used to fight because they don't bind to a foreign antigen. Antibodies are also called immunoglobulins. An immunoglobulin is made up of 4 peptide chains, 2 identical light chains, here, and 2 identical heavy chains, all the way up like that. Each immunoglobulin has 2 identical antigen binding sites, right up here, antigen binding sites. There are 5 types of antibodies. They all bind antigens, but they each have a specialized function. Ig, which is immunoglobulin M, G, A, D, and E. This one is found on the surface of B cells. This is a main blood antibody. This is secreted into saliva, tears, and mucus. This one's found on the surface of B cells, and this one fights parasites. When B cells bind to a foreign antigen and get signals from the T helper cells here, they become activated. Activated B cells begin to make large amounts of increasingly effective antibodies. That's these little Y-shaped people over here. Okay? Because the B cells are also dividing very rapidly, they can produce a great quantity of pathogen-fighting cells. Some more antibodies. And of course, these are the activated antigen-specific B cells. The B cells that make the most effective antibodies may continue to mature into cells that provide specificity and memory, the special features of the adaptive immune system. B cells may differentiate into large plasma cells devoted to secreting high-affinity antigen-specific antibodies during an infection. Plasma cells are like small factories. Or they might become memory B cells. Those are surveillance cells that are long-lived and protect you from the same pathogen for years to come. Memory B cells, kind of like security guards. Clones of the activated B cells that are making high-affinity antibodies, they'll differentiate or mature into cells with different functions. The plasma cells, which will continue to secrete the antibodies, and memory B cells, they will display high-affinity surface antibodies on their surface there and circulate. One way to measure a patient's adaptive immune response is to measure the number of antigen-specific antibodies in their blood. The first time people are infected with a pathogen, it takes 7 to 14 days to produce an antibody response. The second time a person is infected, the immune response is faster and stronger thanks to the memory B and the T cells. So here's our concentration of antibodies, and here's time. So for the first exposure, it takes a while before it gets going, and then it doesn't get very high. If you get hit again with the same pathogen, watch this. It responds almost immediately and much higher, creates many more antibodies. Antibodies work in 2 general ways. If they bind to antigens on foreign invaders, they may neutralize them or help kill them. The antibodies neutralize pathogens by coating their surfaces so the pathogens can't invade cells or carry out life functions. Or they can help kill the invaders in a process called antibody-dependent cell cytotoxicity, or ADCC. The same picture we had earlier, just some more details on it. So they're large proteins with many domains or substructures that can bond to other proteins and cells. One end is the antigen-specific end, and that binds tightly to foreign antigens. Foreign antigens can be found on the surfaces of bacteria, parasites, viruses, virus-infected cells, and some cancer cells. At the other end are domains that can bind receptors on neutrophils, macrophages, natural killer cells, and eosinophils. These are all white blood cells that kill target cells when activated. The antibody protein serves as a bridge that brings pathogens and killing cells together and then activates the killing cells. So here's our antibodies. The killing cells destroy their targets in different ways. Macrophages and neutrophils are phagocytes that kill by engulfing targets. Eosinophils and natural killer cells release a burst of toxic chemicals that bust holes in the membranes of pathogens. So here's our pathogen down here. The antibodies are bound to it. The killer cell then binds to the antibodies. Here's our killer cell. It releases toxins. You can see you got all these little blue things. They're out here now. And that busts through the memory of the pathogen, and then we have a dead pathogen. Here's a summary of all the cells we've talked about. We have natural killer cells. They recognize common antigens produced by cancer cells and virus-infected cells and kill them with a chemical burst. Neutrophils phagocytose bacteria. That means they engulf them. Here's a bacteria in there. And anything coated with antibodies. Macrophages also phagocytose bacteria and cell debris at sites of inflammation. They present antigens to T cells. They are a bridge between the innate and adaptive immune responses. Dendritic cells present antigens to T cells inside lymph nodes. They are dedicated APCs. And some more cells. Each T and B cells makes a unique antigen receptor, displays it on the surface, and participates in surveillance. T cells make TCRs, while B cells make antibodies, also known as immunoglobulins. T helper cells release lymphokines that activate other white blood cells when a TCR is bound by a presented antigen. T cytotoxic cells kill targets by releasing toxic chemicals when a TCR is bound by a presented antigen. Plasma cells are specialized B cells that secrete large amounts of antigen-specific antibodies into the bloodstream. And finally, memory cells. They are long-lived B and T cells that previously responded to a pathogen, and they remain in circulation, providing immunological memory.